Okay, good. So, yeah. Do you want me? No, I wait a minute or I...
Wait just until we hit 9 o'clock. So I'll let you
Okay.
know. All right, you can go ahead.
Okay, so good morning there, good afternoon here in Europe. I'm Raquel Ponti, and I'm a postdoctoral researcher, Marie Curie postdoctoral researcher at CBO in Portugal. And I'm gonna present you this work that is part of this postdoctoral period that is called Morphological Evolution on Island Birds. So most of you probably know about the island features, but I'm gonna do a short summary. So islands usually are small, have small areas, and as the name says, they are isolated areas as well. And they have some particularities, for example, that some of them, or most of them, have limitations in their resources. And they used to have also milder climates compared with the continents. And altogether show also normally low species diversity. So all these characteristics are common on islands and are particular that make species to adapt to this kind of environments. So when the patterns of when there are common patterns of adaptation on islands, we can call them the island syndrome, and we know different common patterns like morphological patterns, behavioral, life history traits, or signaling. I can give you some examples. For example, the most known common pattern of adaptation on islands regarding the size is the island rule, where small species that from the continent, when colonized an island, became bigger, and big species from the continent, when colonized an island, became smaller. But there are many other patterns, and here I'm going to explain some of them regarding the morphology. But there are others, like the behavioral ones. We know that usually species on islands tend to be less aggressive, or or regarding the signaling, for example, it's known that at least in birds, birds on islands tend to be less colorful than continental ones, probably because the signaling change in the and the patterns between the behavior between competitors and intra-specific signaling change on islands compared with the continent. So here I'm gonna focus on the morphology. And one of the main hypotheses here that I want to test is that because of the reduced area and resources on islands, plus the less number of competitors and predators on islands as well, we expect that birds reduce their flight capacity and tend to be as well more generalistics regarding the habitat exploitation, because they increase their terrestrial behavior. So they tend to colonize terrestrial habitats as well as the aerial ones, the trees. So they broaden their niches. So as we expect this, we expect as well that the morphology changes on birds. So we wanted to test basically three morphological aspects of birds that are the wing shape. Here we are going to call it a hand wing index, and I'm going to explain what is this. Basically, the hand wing index is an index that shows you the shape of the wing. So that goes from more rounded to more pointed. So bird species that tend to migrate or fly a lot tend to have more pointed wings, and species that are more resident or for example on islands tend to have more rounded wings. So this is what we expect. We also expect to find shorter wings compared with the continent, and increase their tarsus length because we expect that species on island have less flight capacity and became more terrestrial. We have other hypotheses with other variables here. For example, the latitude. We expect that birds decrease their appendixes with the latitude because of the Bergman rule. You probably know that species where they are in colder areas tend to have shorter appendixes to better thermoregulate. So we expect as well that birds have shorter wings and shorter tarsus with the latitude. And we expect more pointed wings with the latitude because most of the birds that are in high latitudes are migratory and then fly more than the sedentary ones that tend to be in the tropical areas or temperate areas. So then we have other hypotheses regarding predation. We think or we expect that if a species is exposed to a high predator pressure, birds tend to need to escape from predators, so it became less terrestrial, and then they should have more pointed wings, more longer wings, and shorter tarsi because they are less terrestrial. And regarding competitors, as I told you before, species, for example, on islands tend to broaden their niches because they on islands there are less competitor pressure in theory. So we expect as well that in presence of competitors, this increase of tarsus is not going to happen because they don't explore these new habitats in the ground. So to test all these hypotheses that we have here, we used 772 pairs of island and mainland bird species. So basically we used 700 and something endemic birds from islands worldwide, as you can see here in the map. We used only endemic species that occur in islands that are smaller than 11,000 kilometers. So we wanted to just focus on the small islands. And also we only used islands that are oceanic, or at least that were not connected during the last glacial maximum with the continent. So that are basically isolated from the continent, and they were isolated as well in the past. So this is for the island species, and for the mainland species we used the most recent, well, the sister species from the continent that inhabited similar latitudes. So in total we have 1,144 different species from different orders and families to test all these patterns around the world. So as I just explained before, we use these three morphological measurements to see what happened with the change of the shape of the bird. So we use the wing length, the wing shape, and the tarsus length. And to account for predator pressure, we use the number of raptors that predate on birds only, that occur with in the same island or in the continent with the species that we are studying. And for the competitor pressure, this was more tricky because it's very difficult to account for competition. But as a proxy, we used the number of species from the same family that cohabit as well with our focal species, because we expect that species that share, from the same family share some characteristics, ecological characteristics and morphology as well, that made them potential competitors. So we perform different Bayesian models. Basically, we did two sets of models. One were the simple models, where we only wanted to test if there is an effect of insularity. controlled by body size, body mass and latitude. And then we did other set of models, complex ones, that we included the predator and competitor pressures to see if there is an effect. And we repeated all these models using all the birds that we had, and also separate models only using passerines, because we expect that passerines tend to be very flying species, and we expect that they will suffer a bigger change when they colonize islands instead of other species or other families. So here there's a lot of numbers, so you don't need to focus on them. But basically here where we have the simple models where we only explore the insularity, and what we saw is that species that there was an effect of insularity on the shape of the wings on birds, using all the birds and also using only passerines. We didn't, so in this case, as it's negative, we saw that birds on islands have more rounded wings than the continental ones. We didn't find any effect on the wing length, so basically the wings were similar from island to mainland. And we found as well an effect of insularity in the tarsus length, where we saw that species in the islands have longer tarsi. So having this, we wanted to explore the other variables, and we only focused on the wing shape and the tarsi because there was the only ones that have an effect in insularity. So regarding the wing shape, we included the predation because it's for the one that we have an hypothesis. And as we saw in the simple models, we have, in this case, it was not significant, but we can see that there's a tendency, but there was an effect, a significant effect in the simple models. So we can see that insularity has an impact in the shape of the wings, so more rounded wings on islands. But we didn't find an effect of predators, not in the mainland and not in the islands. So it seems that the shape of the wings is more related to the loss of capability of flying on islands, but not because of the predators, but because of the reduced area or other variables. And then we explored the tarsus length, and again we found that actually yes, there's an increase of tarsus length on islands, both using all birds and passerines. But this increase of tarsi is no longer, or is less evident when there are predators on the island. So because we can find here that this is negative. So when there are raptors on islands, the tarsus decrease. So, and also this only happened on islands, not in the mainland. But for example, for competitors, we found the same effect, more or less, yes, in both mainland and in the island, at least in passerines. So in presence of raptors and in presence of competitors, species tend to shorten their tarsi. So in summary, what we can see here, what we can conclude is that the wing length doesn't change between insular birds and their mainland sister species. That what changes is basically the shape of the wing, so they become more rounded, and it could be related to a reduced fly activity on islands. That there's no effect of predators or competitors in the wing shape. That the tarsi increases on islands, probably because the species become more terrestrial, and especially when there are not predators on the island, because we have both islands with predators and without predators. So, and finally, we saw that longer tarsi on islands may indicate a larger variability of habitat, or exploitation of larger habitats. So, but basically when there are not competitors. And yeah, and that's it. I want to thank you for your attention and just present you the collaborators. And if you are interested in more projects, you can check the website. Thank you.
All right. Unfortunately, I don't think we're going to have time for questions.
Oh,
So we're
yeah.
probably going to have you stop sharing your screen so our next
Oh,
speaker
sure.
can do so.
Okay. Sorry.
And so then, Lucrezia, do you want to share your screen? And you can begin.
Okay. So good morning, everyone. And I'm going to present part of the results of my PhD project, which was focused on the mechanism underlying diversification on two different ecotypes of the genus Dianthus within the Mediterranean region. We know that flowering plants have been subjected to a huge diversification, with some groups being experienced particularly fast radiations. And one of these example is the genus Dianthus, and in particular the Asiatic clade of this genus, which has been reported as the fastest radiation documented in the flowering plants. However, the mechanism underlying this radiation are still poorly investigated. And we have just few examples of groups within the Asiatic clade that have been studied from an evolutionary and adaptive point of view. What we already know about this clade is that within one of this group, that is Dianthus broteri complex, the evolution of different ploidy level was a key driver of diversification. But this was probably an exception because most of the species of the genus Dianthus have the same ploidy level. So this suggests that different ploidy level were not the key driver of divergence within this group. Recently, a study conducted within an homoploid complex of this clade, the Dianthus sylvestris group, identified the climatic factors as the main drivers of divergence that lead to a range shift between the three main lineages and the differentiation among these lineages. However, these are just few examples, and there is still so much to investigate within this clade. For instance, we still don't know what can be the main drivers of divergence, and in particular, if biotic factor has the potential to drive this divergence. We also don't know what happened in the early phase of divergence, and if reproductive isolation is able to fastly evolve within this clade. But in particular, we don't know what happens in secondary contact, because all the groups that have been already studied were focused on allopathic populations, and we don't know what happens when these different lineages came in contact again, and if we can have still gene flow in the early phases of divergence. So to try to answer this question, we focused on the complex of Dianthus rupicola. That is a particularly useful model because it includes 5 different ecotypes that are different from a genetic and a phenotypic point of view. And in particular, it includes 2 ecotypes that occur in allopatry, but also in close proximity. And the distribution of this species is highly fragmented. So different populations are exposed to different selective pressure, making the study system particularly useful to study local adaptation and the mechanisms underlying diversification. And also this group has experienced particularly recent divergence because one of the 2 ecotypes evolved on the volcanic archipelago of the Aeolian Islands. That is a quite recent formation. So the divergence didn't occur before 270,000 years ago. And this, of course, makes this system particularly useful because we can study what happens in the early phases of divergence. So using this study system, we try to understand, first of all, when and how the 2 ecotypes diverge using genetic data and performing analysis to understand the demographic scenarios. Then we try to understand if these genetic differences could also lead to reproductive isolation between the 2 ecotypes, and also which are the putative mechanisms underlying this reproductive isolation. Then we try to understand, after having estimating the genome sites between these two ecotypes, if there is an allometric relationship between genome sites and floral traits by characterizing floral traits in a common garden experiment. And then we try to understand which are the drivers of this divergence, the ecological drivers of this divergence, characterize the biotic interactions in natural population, the floral traits, of course, in this population, but also estimating the reproductive success. So first of all, we found that these two ecotypes are actually different from a genetic point of view. But interestingly, we also found that there is a sympatric population where pure individuals of the two ecotypes co-occur without mixing. We also found, as expected, that the Aeolicus ecotype, the insular one, evolved from the ecotype that is located on the mainland, and that this divergence is present. But now we asked if this genetic divergence could also lead to reproductive isolation in a short period of time. And what we observed by conducting reciprocal experimental crosses between different populations of the two ecotypes is that by collecting fruit at different time intervals at 3, 6, and 9 days after pollination, we observed that in the interecotype crosses, 9 days after pollination, the embryos start to degenerate. And this is due to a dysfunctional development of the endosperm. We also collect these crosses after 10 days. And what we found is that these seeds, the one produced from interecotype crosses, are not able to develop, and also none of them is able to germinate. This kind of barrier has been reported also in other species of the genus Dianthus in classical studies, but the mechanism underlying this reproductive isolation has never been really investigated. So we try to understand, in our cases at least, what can be this mechanism. And to do this, we estimated, first of all, the genome size over this population of the two ecotypes. And surprisingly, we found a strong difference in the genome size between the two ecotypes. At this point, to understand if there is a causal relationship between genome sites and reproductive isolation, we correlate the genome sites of all the crossed individuals to the percentage of seeds developed. And what we found is that there is a strong association, and the differential parental genome dosage, the higher is the difference, the lower is the percentage of seeds that are able to develop. So once we observe a strong association between genome sites and reproductive isolation, so suggesting that this is one of the mechanisms explaining this isolation, we also try to understand if genome sites that can affect plant phenotypes, because it can determine the number and the dimension of the cells, if it's correlated to several floral traits in our study system. So to do this, we characterize a lot of different floral traits in natural populations and in a common garden experiment. And as we expected, there is also a clear difference in phenotypic traits. But among these traits, style length appear to be strongly associated to genome sites. So the higher is the genome sites, the higher is the value of the style length in both the ecotypes. And this is one of the traits that show the highest divergence between the ecotypes. So we try to understand if this divergence was driven by the selection imposed by different biotic interactions in the natural populations. So first of all, we try to understand if this trait was shaped by the action of different pollinators. But what we found by analyzing different populations of the two ecotypes is that in both of them, Macroglossum stellatarum is the main pollinator, and that overall there is no significant difference in the pollinator community composition. So we try to characterize also the antagonistic biotic interaction to understand if they can have a role in shaping this divergence. And what we found is a clear pattern in this kind of interaction, because in the Rupicola ecotype, we found that most of the populations had an high percentage of predation by the nursery pollinator Adena, while all the populations of the Aeolicus ecotype show the absence of this interaction. So we try to understand if this divergence was driven by a divergent selection imposed on a population of these two ecotypes. And what we found is that style length in the populations of the Rupicola ecotype is negatively selected, since the higher is the length of the style, the higher is the percentage of predation by Adena, and the lower is the reproductive success. So it means that in these populations, the flowers that are smaller, and in particular that have a lower style length, are also the one that experience the highest reproductive success, because this trait make them this is able to make this population escape from the predation by Adena. While the opposite trend was observed in the populations of the Oligos ecotype, where style length is positively selected. So flowers that are bigger and at a higher length of the style are also the ones showing an higher reproductive success. And this could be related to the absence of Adena, so they don't need to escape from these antagonistic interactions. And being bigger also make them more attractive to the main pollinator, that is Macroglossum stellatarum, that of course is attracted to flowers that are bigger and that produce more nectar are the ones that are more attractive for this kind of pollinator. So in conclusion, what we can say is that in this system, the divergence was particularly recent compared to all the divergence events that happened within the clade, and that biotic pressures seem to have played a key role in shaping the diversification by driving the phenotypic differentiation between these ecotypes, and in particular in a trait that has an allometric relationship with genome sites, that is potentially the key driver of reproductive isolation, and thus boosting this difference in genome sites. They were able to make reproductive isolation, strong reproductive isolation, evolve in a short time. And also, what you can say that by since we found sympatric populations where the two ecotypes are able to co-occur without mixing, that even in a short time, reproductive isolation is strong enough to keep the two ecotypes separate. And this does suggest that the most plausible scenario for this complex is that the diversification will proceed and maybe will have the formation of two different species. Thank you for your attention and for the ones that collaborate to this project.
Right, so we've got plenty of time for questions. If you want to ask a question, use the reaction of raise hand. It'll bring you to the top of the list. We can get you to unmute and ask your question. All right, if there's no one who's going to start with one, I'll start out with asking you, so what do you have planned as follow-ups for this?
Sorry, I didn't hear.
What do you have planned to follow up on this? What would you like to do next to extend the study?
Okay, this is a particularly complex system because the biotic interaction that we found in natural population are actually really complex, because it is a species that flower in summer in the Mediterranean. So is probably the only one that we can find in August in southern Italy. And so it attracts a lot of different interactions, so biotic, antagonistic, and mutualistic interactions. So what we found here is that maybe Adena is the one that has a significant effect on phenotypic traits and all the things that I described here. But we also think that maybe other interactions can have a role in shaping this divergence, because we have, for instance, a lot of different parasitoids that are related to the presence of Adena. And so we don't have just an antagonistic interactions, but a really complex network. So our aim now is to, first of all, characterize better all these interactions and understand which is the role of the different interacting insects, but also understand better which is the role of genome sites. Because for now, what we found surprisingly is that this barrier is really strong. because by performing thousands of experiments by comparing populations of the two different ecotypes, none of the seeds that were produced were able to germinate. So we didn't expect this. We thought there's some extent of reproductive isolation, but not to this strength. And so our aim is to understand how fast the evolution of genome sites and the difference in genome sites evolve, and in particular, which is the region of genome sites that is present in the one ecotype and is absent in the other one, and which is the role of these regions that are absent in the mainland, to understand if these can explain somehow the difference also in the biotic interactions and in the phenotypic traits.
Thank you. I'm going to ask you to stop sharing your screen now so we can get the next person up to get ready for their talk that starts in 20 seconds. So Bhanu, can you share your screen?
Yeah.
And actually, yep, there we go. So you're not yet sharing anything. There should be a large green button in the middle of the bottom of the Zoom controls.
Oh, yeah. Can you see now?
Yes, we can. So you can begin your talk.
Yeah. Sorry. Yeah. Hi. Hello, everyone. So at first, I'd like to thank Evolution 2025 for the platform to discuss the science that we are currently doing. So I'm Bhanu. I'm working with butterflies at Indian Institute of Science, Education and Research, Thiruvananthapuram, India. And today I'm going to discuss one of the work that we are currently doing. Now, hello.
So you'll need to advance to the next slide. So you can do that from your PowerPoint.
Okay. Yeah. So you can see the next screen, is it?
We're still seeing your title slide. There you go.
Yeah. Okay. So yeah, again, sorry for that. Yeah. So insects, like any organism, lives in environment where there are a lot of stressors around. Predators, parasitoids, that could be there could be intraspecific competitions. Biotic factors could be temperature, desiccation, other things. So in this talk, two aspects, one predators and one desiccation is what I'm going to touch. Now, there are multiple predators that feeds on insects that we are aware of, and there are some factors that makes insects vulnerable to desiccation. And one of the factors is their body size. They are small. Surface area to volume ratio is high, hence the amount of water that comes out of the body are high. And the amount of water that comes out of the body depends on the ambient condition. If the ambient condition is dry, then the probability of water that coming out of the body higher. So they can desiccate, the insects can desiccate. Now, these stressors, be it predators or be it desiccation, this stresses, they varies across different life stages. Insects like butterflies have different life stages, right? So the egg, larvae, pupae, and the adults, these different life stages differ in terms of their ecology and the physiology. For example, eggs are small, tiny. They can't move on their own. So they can't behaviorally avoid predations or any of the environmental stress. Whereas larvae and the adults, larvae are relatively mobile than the eggs. larvae and the adults, they can behaviorally avoid getting predations, and also they can avoid some environmental stresses like desiccation or heat. Similar to egg pupae, again, pupae also are immobile, so they can't behaviorally avoid the predation and the stresses. So in this talk, I am focusing on the pupae. So there are multiple strategies that evolved over a time that helps pupae to avoid detection by the predator, hence predation. And one of the strategies that we know now is this pupa color plasticity, wherein the pupa color, the color of the pupae, depends on where larvae eventually go and pupate. For example, in this case, this Catopsilia pomana, the butterfly species, if larvae pupates on a background which is brown or darker, then they tend to be, the pupae tend to be darker. And if they pupate on green, the pupae tend to be greener. So such example we can see on the other side of the screen as well. Here, if larvae pupates on the brown background, they become brown, and if pupates on green background, it becomes green. Such phenotypes are seen in many other butterfly species, and it has been hypothesized and tested, and it seems to have effect in avoiding the detection by the predator. Hence, this is one of the strategies to avoid predation. Now, there's another butterfly species called Eurymia blanda. This butterfly species also show the pupa color plasticity. So all these pictures that you see here, these are the pupae of the same butterfly species. And it has been hypothesized that this brown pigment here is the melanin pigment. And looking to the biology of this butterfly, particularly the pupation stage, what happens is when the larvae are done eating, when larvae have finished eating and they are wandering to find the place to pupate, if they pupate on these defoliated twigs, which larvae themselves have eaten previously, if they pupate on these defoliated twigs, then they tend to be browner. And if they pupate under the leaves like this, then they tend to be greener. Now, since this defoliated area, they are directly under the sun, and hence it is expected to have higher desiccation stress. While this pupae that lives under the leaf, that pupate under the larvae that pupate under the leaf, they tend to have the shade environment, and hence probably because of the physiology of the leaf, for example, when leaves are transpiring and all. So because of that, there can be a bit of humid condition, hence low desiccation stress. So we wanted, we had a question: do this browner pupae that's generally form that generally we find in this defoliated twigs, are they having higher desiccation tolerance than the green ones? Now, to test, to answer the question, we came up with a hypothesis. We hypothesized that the browner pupae will have higher desiccation tolerance than the greener ones, and this is our prediction. So what we predicted was the browner pupae. When the pupae are exposed to the desiccation stress, then the probability that the browner pupae will do better in terms of successfully coming out as a butterfly is higher if they are browner in color than for the individuals which are greener in color. Similarly, what happens when there is no desiccation stress as such? So we expected that if there is no desiccation stress, then the color of the pupae and the eclosion success will not have any relation. Now, the possible mechanism why brown pupae might be doing better than the green ones under desiccation stress has been hypothesized and tested in the other insect groups like Drosophila, where the pigment melanin seems to have some hydrophobic nature, hence it known to resist some water loss. Hence, the browner individuals having melanin pigment on the cuticle seems to do better than the greener ones. So to test this hypothesis, we did experiment. We reared the larvae and we exposed them on a greener brown cylinder, and we got the pupae, and then we exposed those pupae to the desiccation stress. And after exposing them to desiccation stress, we recorded whether the pupae that were exposed to desiccation, they will successfully eclose or not. Similarly, there are other set of pupae that were reared in the similar fashion, but they were not exposed to the desiccation stress. We simply recorded whether they will close or not. And to quantify the color of the pupae, we took the photograph and we calculated the R, G, and B, that is red, green, and brown, red, green, and blue. using the custom code using the image, and we calculated the luminance of the pupae. Now, what we found. Okay, so I have done two experiments at different RH systems. So when the larvae that were reared at 85% RH, when the pupae that formed from those larvae, when they were exposed to desiccation stress, what we found was the pupae which were browner, the browner pupae had a higher probability of surviving than the greener pupae. So, but when we when we plot this correlation between the luminance of the pupae and the water loss, that is the amount of weight loss. Water loss here was measured as the weight loss. It's gravimetrically measured, and weight loss during the desiccation assay. We didn't find a strong correlation. So this is another experiment where the larvae were reared at different RH. Here the RH at which RH here is relative humidity. That was 60% RH, and here also we saw a similar pattern that we saw for the previous case. Similar to previous case, here also the correlation between the luminance, which is the proxy for the color of the pupae, and the water loss was not strong. So when we check the relation between the luminance, Pupal luminance, and the eclosion success in the absence of desiccation stress, then we didn't find any relation between the pupal color and the eclosion success. So here the y-axis is like the closed means here large number of individuals, when they were not exposed to desiccation, all of them successfully became butterfly. They successfully come out as a butterfly. So this was for 85% RH reared larvae, and the another set which was reared at 60%. In that case also we see a similar pattern. So it seems that the pupal color has effect when pupal color has some adaptive values when they are desiccated. So this pupal color has been tested to have adaptive values with respect to avoiding predations by avoiding the detection. The pupae on brown backgrounds matches the backgrounds, and thus helps it match and avoid the detection. In this study, in the current study, we could see that pupa color might also have a role other than avoiding predation, and that could be avoiding the desiccation tolerance given the biology of this butterfly. So with this, I'd like to thank all the people that are involved, and thank you.
All right, so we have plenty of time for questions. As a reminder, you can either use the raise your hand function, or you can put something into the Q&A and we can get to questions that way. We have a question. Let me ask you to unmute. All right, go ahead.
Hi, it was a very interesting talk. I was wondering if the caterpillar have some, I don't know, behavior where they seek to form a chrysalis under the leaves if the conditions, maybe the temperature is very high or the humidity is very low in the air. Or,
Yeah. So yeah, the behavioral experiment I'm yet to do, but certainly that experiment would be very interesting to look into. Yeah. I've not yet done the experiment on that part yet.
All right, so Sophia, we also have one from you.
Hi, thank you for sharing. This talk was so interesting. I've never heard about this kind of color plasticity. I was wondering what the mechanism is for how the caterpillars, I guess, know whether to turn green or brown. And if you put them on artificial colors, what would happen in that scenario? Thanks.
Yeah. So how do they know that? I think we are still trying to figure it out. But what we know is they have a sense, like they have a sensory hairs on their skin, which sense the light around them, and based on the luminance primarily, luminance of the substrate on which they are there, they seems to decide. I mean, they seems to turn on or turn off the production of the genes that are responsible for making the pigments that eventually will give the color for the color of the pupae. Yeah. Yeah.
So I will also let you know you've got a question in the Q&A. I'll ask you to stop sharing your screen so our next person can, but feel free to either write a response or give it verbally.
Sure. Thank you.
So Tarunkishwor, you are our next speaker. Do you want to share your screen?
Hi. Yeah, let me share my screen.
All right, and you can begin whenever you're ready.
Yeah. So my screen is visible. Okay.
Yes, your screen is visible. Go ahead.
Yeah, so I'm going to be talking a lot about butterflies now. So I'm going to be talking about a work that we did on the evolutionary pattern of a fascinating set of characters in butterflies, which is popularly known as false head. Before I start talking about the work, I would like to acknowledge the people who have helped me during the completion of this project. So we all know that the animal world is the diversity of colors, and the function of the diversity of colors in the animal world is fascinating. For example, there are multiple ways in which animals can use their color to blend against the background so that they are not detected by the predators. In their cases, they can use this conspicuous coloration to serve as a warning coloration. And in some species, they use the lack of color to blend against the background through transparency. In butterflies, this deflection is one of the most widely studied anti-predatory strategy in butterflies. The eye spots in butterflies is a well-known example over here. And in the group of butterflies, there's another very interesting set of traits, which we will be talking about in more detail. This area, the modification in this area at the posterior end of the hind wing, which is popularly known as the false head, is this type of butterflies is very widespread in two families in butterflies, which is the Lycaenidae and the Rheodinidae. So what constitutes a false head in a butterfly? There are many traits, as you can see here. Using the published definitions, we refine the definition of what constitutes a false head. So it can be the multiple lines running across both the wings and converging towards this region, which is known as the false head region. And there can be these dark spots, which can function as reduced eye spots. And then there are these protrusions at the end of their hind wing, which has been hypothesized to look like a false antenna. And then there can be modification in the shape of the hind wing, which has been hypothesized to look like the shape of a head. And on top of that, in this region, they can have these conspicuous contrasting colorations. So how do these false head traits work? One of the most debated hypotheses about the false head is that the false heads deflect predator attacks. Predators are attracted to this false head, and their attacks are diverted towards this false head area, which is a less vulnerable area compared to, for example, the body or the real head. But there are multiple other hypotheses about how the force head trait may function. Another hypothesis is the intimidation hypothesis, which says that the predator is alarmed by the presence of this force head, causing the predation to stall their attack, thus providing the butterfly ample of time to escape the attack. In some hypotheses, the force head has nothing to do with how the predator interacts with the butterfly. In the case of this anti-force head hypothesis, the predators are directed away from their true head by the presence of this bright conspicuous coloration at the posterior end of the hind wing. So it has nothing to do with how these traits either look like a false head or not. Just the presence of this bright conspicuous colors is enough to misdirect the predator attack. So what are the evidences for this false head hypothesis? One of the earliest and most nicely done experiment on this false head was done by Worms and Wasserman. What they did was they used dead butterflies, and then they manipulated the wings of these dead butterflies to attach either a single false head trait or pairs of false head traits. And then they divided this butterfly body into three sections: the front section, the middle section, and the hind section. And then they looked at the predator interaction, how the predator attacks these butterfly models, and looked at where do they attack them. So let's look at this top plot. This is what happens when the bird first makes the attack contact point. So on the bottom, on the x-axis, you can see what the butterfly model has been. So as we go from the left to right of the x-axis, the butterfly model has more and more false head traits. And if we look at the first attack contact point, the presence or absence of the false head trait does not bother much where the first attack contact point was being made. In fact, most if they have less and less false head traits, most of the attack goes towards the head. But the more interesting thing happened when the bird was handling these butterfly models. So if the butterfly model has more and more tails, if we look at over here, then the handling area goes towards the hind wing area, which is represented by the gray color here. So it gave some sort of support that, yes, when the predator is interacting with these butterflies which have false heads, the attacks are being misdirected towards the false head area. Another sort of like indirect support came from a study from Mexico, where they looked at a subfamily of false head group of butterflies. They studied a lot of thousands of these museum specimens, and then they counted the number of false head traits possessed by each species. So when these butterflies rest, the false head traits are visible when the butterflies rest, and they rest by closing their wings, and the underside of the wings are visible. And if the butterfly species has symmetrical wing damages on the hind wing area, it is sort of regarded as a proxy for an attack has been happened. So if the attack has happened in this false head area, then it is served as a proxy of the attack being deflected towards this less vulnerable false head area. What the study found is that, so here on the x-axis represents the number of the false head traits the species has. On the y-axis is the proportion of deflected attacks. So if the butterfly species has more and more number of false head traits, and the more and more successful deflected attacks happened. So that means basically that if the false head traits is more complex, then the successful deflection increases. So from this, what we wanted to ask was, if the deflection hypothesis is true, then we expect the false head to evolve from a simple to more complex and more elaborate traits. And if the false head traits function together, not as singular traits, if they function together as an adaptive constellation, then we should expect these traits to evolve in a correlated pattern. And we also wanted to explore what was the evolutionary sequence of these false head traits. to do this analysis, we first begin by building the phylogenetic tree, and due to a lot of missing data in the missing molecular data, we adopted a constant tree building approach by using a backbone derived from Kawahara's paper. And in our tree, we have used 900 and around 928 Lysinidae species, which are distributed globally. So when we reconstructed the ancestral state of this faucet traits, we recovered. So to do this, we first built the faucet traits data for all the species represented in the phylogeny. So we relied on a number of online databases to extract the data if the species has the faucet trait or not. For example, if the species does not have a faucet trait, we represented it as 0, and if it has the trait, then we represented it by 1. For example, in the case of this hair-streaked butterfly, it has all the faucet traits, so we coded it as once for all these traits. So when we built the ancestral state of these faucet traits, we recovered that all the faucet traits had multiple gains and multiple lows. So these traits are highly labile. One probable reason for these highly labile traits may be because of differences in the predation selection pressure, or the butterflies are getting different predator community. And to look at the correlated pattern of evolution across these faucet traits, we built Pages and hidden markup models between the pairwise traits. To briefly summarize, we had independent model which assumes that the trait of interest evolve independent to each other, and then we had dependent model which assumes that the trait of interest evolved in a correlated pattern. And what we found was that for false antenna and the spot, the pattern was correlated. Similarly, we found false antenna having a correlated evolution with the conspicuous coloration, similarly with the faucet contour. So all these 4 traits, false antenna, spot, contour, and conspicuous coloration, they evolve in a correlated pattern, which kind of suggests that they evolve through a common selective force towards a functional association, which comes back to our earlier hypothesis that these traits, they together form an adaptive constellation, and it does, our study does provide support for the idea that these traits are together, working together to deflect predator attacks. On the other hand, our results do not provide support for the anti-faucet hypothesis, which states that the presence of the contrasting color alone is sufficient to misdirect predator attacks. The faucet traits, there are 4 traits which showed correlated pattern, but the convergent lines did not show a correlated pattern with the rest of the faucet traits. It's possible that these convergent lines are functioning as disruptive pattern so that the butterfly can first employ camouflage, and when the predator approaches further closer, then the rest of the traits started working as a deflective signal. What's happening at the broader scale? So all this study was done in Lysinidae family. To look at the broader scale, we used a tree which represented 98% of all butterfly subtribes, and we found similar pattern. The 4 faucet traits over here, they evolved correlatedly, and the convergent lines did not show correlated pattern of evolution with the rest of the faucet traits. To look at the possible, the likely evolutionary sequence of these traits, we conducted phylogenetic path analysis by building all the possible 120 path models. These are some of the examples of the path models that we have built. And what we found was that the best path goes from conspicuous coloration to false antenna, spot, faucet contour, and convergent lines came last. and there are multiple studies which shows that there's a correlation between the body size and the conspicuous coloration, meaning that the smaller prey with the smaller body size, they get more beneficial when they employ camouflage color, and if the prey is more larger, then conspicuous coloration can be more beneficial. So we tested if the body size affected the evolution of the false-head trait, and if so, and the rationale behind this test was that if the butterfly is small, then having the false-head traits will make it easier to detect. So it will be less beneficial for them. Assuming that, we expected a relationship like this with larger butterflies having more false-head traits. However, we did not find any relationship between the wingspan and the number of false-head traits that the butterfly species poses. Some of the likely scenario for why we found this result is probably because the most important
I apologize.
factor when
I'm gonna have to cut you off here. You've run out of time, and so we need to move on to our next speaker.
okay.
We're gonna just wait until we get right to the 15 because people are moving back and forth between the different sessions.
Okay. Can you hear me well?
Yes, we can.
Okay, that's nice.
All right, you can get started whenever you're ready.
Okay, thank you very much. Hello, everyone. I hope you're having a great day. My name is Victor Araújo. I am a PhD candidate in the Universidad de los Andes in Colombia. I am right now doing an internship. I am like a visitor fellow in Universität Bern and the Naturhistorisches Museum Bern in Switzerland. I'm a SCAS fellow. So I'm finishing my PhD basically in Switzerland, taking some data to have a new chapter. And I'm talking today about the elevational patterns of morphological evolution in Harlequin toads. Sorry, I couldn't change. Okay. So basically, size and shape affect and are affected by the environment in many factors in many ways. So in chordates, for example, we have a huge diversity of sizes and shapes. And here we can see the smallest chordate in the world, that is a small fish from Indonesia. And here we have the biggest chordate of the world right now, that is the blue whale. But trust me that the blue whale is small compared to many species from the past. So we had an even bigger diversity of sizes. I'm so sorry for the Americans for the values in meters because you use the imperial system. But basically, those small fishes are around 0.2 inches, and the blue whale can come to 97 feet. So they are really huge. But not only the environment, but also the evolutionary history play a role in the evolution of size and shape. So for most of the organisms, we see that closely related species have a very similar size and shape. But here we have an exception. So when we see the paleognates, we see that most of them are big and they probably had a big ancestor, but there are two groups that are pretty small compared to the big paleognates. So the kiwis and the tinamous are small compared to the ostrich and the emu and the rhea of other regions of the world. And this is probably because of the role of the environment over the body and the shape of those animals. So basically, in the past, some researchers noticed that there are some patterns of evolution and of well, body size and shape. And Bergman, for instance, noticed that animals with a higher latitude would display a bigger body compared to those close to the equator. So we have some examples, and I brought the example of the bears. So the big polar bears are really big compared to the Andean or the Malay bear, right? And this is due the proportion of surface and volume. So those bigger animals would have less surface comparatively to those smaller animals, and would lose less heat to the environment and would conserve a very stable temperature. In the past, Allen also identified a pattern that basically the appendages of animals in colder places would be smaller than the appendages of those animals of warmer places. So here we have an example for lagomorphs that they usually have longer ears and limbs when they are in warmer longer if they are in warmer places, and these species from the Arctic show a really small ear and a small limbs. What about the amphibians? Well, basically amphibians are ectotherms, so they their temperature varies in accordance to the environment. And for those animals, the range of temperature can vary a lot. So they don't have a very strict range of temperature as the endotherms. And for that reason, many researchers say that the Bergman's rule do not apply for amphibians. But what we see in many papers, if we do small research, is that they basically have different results, right? So in that case, for amphibians, we have a similar situation as the Schrodinger's cat, in which those rules apply and do not apply for amphibians at the same time. So, specifically about amphibians, we have a very interesting and endangered group, the Harlequin Toads, the genus Atelopus. And here we have some examples, some photos of species of Atelopus. And sadly, they are not proportional, so they are in the same size, but you can see a difference in size if you take one and the other and compare them, right? So here, for example, we have those two on the bottom. They are from high mountains. Meanwhile, those in the right, mid, and top, they are from lowlands. So we can see they are slender, they have slender bodies, and if we compare the size of those animals, they are really different. So most of the animals from high mountains are robust and more fatty compared to the slender species of the lowlands. So here's an example. I was measuring some specimens from the Museum of Geneva. Both the species are from Ecuador. They are closely related, but one of them, the one on the right, is from from lowlands, and the one in the left is from highlands. So I had as general objective to investigate the evolutionary patterns of body size in the genus Atelopus by integrating comparative morphology in an elevational gradient, and as predictions that if body size evolution is phylogenetically conserved, then closely related Atelopus species would be likely to exhibit more similar bodies and similar body size and shape related to other species, and that if body size variation is influenced by the elevation, then those animals in the same elevation will show similar body size and shape. And how did I do it? First, I got the information mostly from GBIF of different museums of natural history. So I made a database of those collections and museums of natural history to try to visit as much as I could and to have as a result the best database with most of the species of the genus. And I also checked for the elevation data. For some data, for some collections, we don't find it easily, but in some situations, we can find the location and try to find a value close to the elevation data. It is tricky sometimes because the variation of elevation is real in the Andes, because just moving 100 kilometers, you can have 2,000 difference of elevation, but we try to be the more precise as possible. And as measurements, I had the SVL, the snout vent length, the tibia length, the foot length, the head width, the interorbital distance, the eye diameter, and the sacrum width. I used the phylogeny of Lutus and collaborators of 2025, very recent. The problem is that that phylogeny is not complete, but mostly because the genus is basically extinct. I mean, it's not extinct, but around 50% of the species are possibly extinct or critically endangered. So it's very difficult to find them. And we have some samples from museum. They are being sequenced now to obtain a better version of the phylogeny, more complete with more species. And as it's a statistical analysis, I did simple linear regressions, but I also checked for graphical evolution of traits. I also calculated the phylogenetic signal, and I did a phylomorphospace graph to check visually the evolution of the traits. So basically, we have some preliminary results. It's not all done. I wish it was, but as I have limited resources and time, I'm trying to do my best. So as first result, I got a correlation between elevation and tibial proportion. That's very strong. So basically, we see that each point here is a species, and we have females in red and males in blue. And we see that is a strong correlation between elevation and tibia proportion. And for size, the SVL, the snout vent length, we have the opposite. So those from higher environments usually display a bigger body. For the phylogenetic signal, I had very interesting results for tibia proportion and for elevation. Not so good for SVL, the total body size, but I guess it might be better if we increase if we use the most recent phylogenetic tree that is being constructed. So that's why I didn't use it. because it will have much more species and it will be much more complete. We have good values for tibia proportion and and elevation of Wegel's lambda, but not too well for Blomberg's K. We are not sure, but we have some hypotheses. Probably it's a matter of adaptive selection. And we are seeing something that, I don't know, might be slower than we thought that it would be, but we still don't know. We still have to try to discover what's going on in that case. And for the evolution of the traits, we prepared some trees with every each of the traits that show a significance, except for the SVL that I put here because it's slightly significant. No, it's not significant, but it's very close to it. So we can see that basically the closely related species have similar, in this case, in the top left, we have the SVL. In the bottom left, we have the elevation. So those closely related generally have a similar value. And in the right, we have the proportion of the tibia. And I use the data for females because they show more differences, but for males, it is basically the same. And we here, for the phylomorphospace, we can see some of the different clades of the species of Atelopus identified by colors. And here we have for SVL, so it's not very clear, using only the species present on the tree. But I'm sorry, here it's SVL in the bottom, but actually it is tibia proportion. So here for tibia proportion, we see something very clear. First, as a
I'm sorry, I
correlation.
do want to let you know we've hit 15 minutes, so you might
sorry.
want to wrap up. Since you are the last speaker, we can afford to go a little bit long, but you do want to wrap up here.
Okay, I just need 1 extra minute, I guess. So we see a little bit of a correlation, but also the different clades are grouped basically. And just to finish, apparently we have a strong role of the environment on those animals. We can try to add some climatic data to enrich our results to check if that is only the elevation or temperature, average temperature, and other characteristics are the climatic data to check if they are directly connected to the traits. And we can, well, also I have information from many species. We can also try to help people that are working with taxonomy, trying to separate and identify morphogroups. So that would be very helpful to those people that find a species and don't know for which clade they belong. So I guess I have enough data to have something like that. And that's all. Thank you very much for everyone. And thank you for the collections that I visit and who financed my work. That's it.